Okay, good morning. Welcome to FlowTex Day 3. We're getting started this morning with the advanced technology session. I think we have some really cool talks that are not your average flow that you've seen. So our first speaker today is John Hickey. John received his PhD in biomedical engineering from Johns Hopkins University and is currently postdoctoral fellow in Gary Nolan's lab at Stanford, where he uses and builds systems biology tools to describe spatial relationships between cells and tissues. His work has been recognized with the receipt of multiple awards like the NSF Graduate Fellowship, the ARCS Scholar, Siebel Scholar, Hans J. Prochaska Award, and the American Cancer Society Postdoctoral Fellowship. And John, I saw come and give a talk at MD Anderson a couple months ago, and I thought it was so cool. I almost immediately asked him to come and speak here. So I think it'll be really fun and kind of something new and different. So in his seminar today, John's going to provide answers to how does cellular organization in the intestine change from the small intestine to the colon? How does the cellular microenvironment contribute to chronic inflammation-induced cancers where epithelial cells change from normal to metaplastic to dysplastic and finally to tumor? How do we design cellular therapies with capacity for changing their microenvironment in mind? And finally, how do we make sense of the spatial omics data sets and extract additional meaning? So with that, John, please take it away. Maybe all you.
Thanks, Sarah, for the kind introduction, and thanks for the organizers for putting together this amazing event, and for all of you for being here. Can you hear me well up there? Yeah, okay, great. Yeah, so I'm really excited to talk to you today about some of the work that I've been doing as a postdoc, and doing it at Stanford with Gary Nolan. And so first, to start off, I thought I'd just give you a quick introduction to myself and the research, and then, like Sarah mentioned, these three vignettes of how we're using this technology to really understand cellular organization in tissues, and then stop with some conclusions. So first, who am I? Well, I think that depends on what you analyze about me, right? You can think about me from my generations, six to eight generations back, where I come from, what I look like, 155 pounds, white male. Or a lot of you will judge me based on my scientific output or what I secrete, and so forth, right? So I think it matters, what you analyze about me to define me. But I think it also matters how you analyze that data as well. If you looked at the time that I spend doing different activities, you might say, well, he's a sleeper. If you look at my differentiation trajectory, you might say, well, okay, he was a chemical engineer, now he's a biomedical engineer, he's trending to be more a technologist or immunologist. Or if you lump all the characteristics about me into a low dimensional space and look at the top differentially expressed ones, you can say maybe he's cross-disciplinary, he likes to collaborate, he likes computational techniques, or you might just find out that I like chocolate. So yeah, so I think that this, this aside is kind of fun, but I think that this matters a lot for the type of biology that we're doing, right? And so one other question I will pose to you is, what do you see when you look at this image? And I realize, this is more flow cytometry, and not many, maybe not many of you have seen this. This is an H&E tissue. Basically, it's a slice of tissue taken from a patient, stained with two dyes that allow you to see individual cells within the tissue. And, for me, just one person, I can have multiple perspectives on this in terms of how or what I analyze about just this image. So for example, if I put on my pathologist hat, I see this area of the tissue and realize this is diseased tissue and abnormal, and this is a problem that's potentially cancer for the patient. Whereas if I put on my cell biologist hat, I can start to see cell-cell interactions that may be recurrent across the diseased tissue that might implicate a mechanism into the disease. Whereas if I put on a chemical engineer hat, I see separations of cells within the tissue. And anytime we think about separations within engineering, we know that there's energy going into the system, or a low entropy system. So, that's just three views of this data or image that I'm looking at on the screen. And, if I combine this, I start to think to myself, okay, well, what cell is driving this organization? And, you can think, if you ask someone who studies immune cells, they'll say the immune cells are driving the organization. Whereas if you ask someone who studies epithelial cells or tumor cells, they'll say the tumor cells drive the organization, and so forth, basically with stromal cells as well. But this kind of brings the fourth view of this tissue that I have of, another type of hat, which is a systems biologist type hat. And how I see tissues is really a system of multi-scale network of interactions. And what do I mean by this? So if we take a tissue like cancer, we can think about multiple functional tissue units that are arranged spatially that are accomplishing the overall function of that organ or tissue. And within these functional tissue units, we have individual cellular neighborhoods. So neighborhoods that are cellular organizations that are grouped together by their composition, and they are also spatially interacting with each other. And within these cellular neighborhoods, we have cell-cell interactions where individual cells have protein ligands that are either secreted or on their surface that are interacting with each other. And then within a given cell, we have networks of proteins that are interacting to cause changes in function or phenotype of the cell. And, I think we've done a really good job of measuring all of these things independently of each other. But I think to understand organization and how cellular organization implicates function for large tissues, I think we need to be able to measure these and connect them. And so in the Nolan lab, so I guess before I go to the tech, just another motivation slide. So I think if we're thinking about taking apart something complex like a tissue or a complex Lego set, my kids love Legos, I think we've done a really good job of decomposing it and looking at all the individual pieces and really understanding what pieces we have to work with. I think, now where we're going is, okay, putting these pieces back together into modules or extracting these multi-cell modules from tissues. And so one of the technologies that was developed in Gary Nolan's group was this technique called codex multiplex imaging, which basically you have antibodies, you label them with DNA oligonucleotide barcodes. Basically, each antibody gets a unique barcode. That allows you to stain with as many antibodies as you have barcodes. Usually we're staining in the 50 to 60 range, but you can go up beyond that. And then to image, we anneal the complementary sequence that is attached to a fluorophore. And usually with a microscope-based system that we have, we're doing 4 fluorophores at a time. We image those, then we do a gentle denaturation where we strip those off and then image the next set. And we continue to do this cyclic imaging until we have imaged all of the antibodies. So I've been involved in developing these protocols, but not only, how do we do the imaging, but how do we think about doing the data processing and storage? What metadata should be associated with this at each individual stage? How do we think about these antibody panel developments? When we actually get the data, how do we extract meaning at a single cell level and normalize and do cell type clustering or cell type annotation? And then, how do we think about making this more machine readable so that, multiple labs can use this and multiple databases can use this? So this is a lot of work that I think helped me to achieve good quality data sets and also hopefully for the field to be able to do this as well. I'm not going to talk about these efforts, but basically using these as underlying tools to apply it to the biology that I'll share next. So first, I'll talk about our approach to the healthy human intestine. So what we were interested in is the healthy human intestine is a very long organ. It's nearly 9 meters in length, and it's also diverse in the 9 meters as your digestion flows. And so we thought, obviously there's got to be some cellular organizational differences between the small intestine and the colon. And so we took 4 different areas of the small intestine and 4 different areas of the colon from healthy human donors. And we took them and imaged them with this codex multiplex imaging. And what does this look like? So this is an example image from this codex multiplex imaging. This is only, 5 of the 57 antibodies that we use. Here in the pink, you see the epithelium, which is the part that's closest to the lumen of the intestine. Below it, you have the submucosa, which is a stromal area. You can see a lymphoid follicle within it. And then here you have this muscle area of the intestine. And using this type of imaging approach, we can get to the single cell resolution. We can start to study things like the mature follicles or developing follicles, even things like the immune cells that are between the epithelial cells within the mucosa, or even the base of the mucosa, which is the stem cell niche for the intestinal cells. And we can use many different markers and be able to identify many individual cell types. So how do we start to think about this beyond just pretty pictures? How do we get quantitative measurements? And this is how it comes to be like imaging cytometry, right? So what we do is we apply a deep learning algorithm that draws a little segmenting border around each individual cell. And from that border, we're able to quantify the average fluorescent intensity for each individual marker, or related to the average protein expression from that antibody that we're using in the imaging. And then we can go and do the cell type clustering like we normally do, where we're clustering cells in an unsupervised manner based on the expression of known markers that we would do with normal flow cytometry. But one of the challenges with this approach, as opposed to maybe flow cytometry, is that cell segmentation isn't perfect, and there are some imaging artifacts. And so what it requires is to go back and forth between the fluorescence, the raw data basically, and what we cluster in the unsupervised clustering to verify that our clusters are actually clean and pure and actually represent the cell types that we're looking at. And this process takes, nearly 20 hours a sample. And with this study that we're doing on the healthy human intestines, that was going to be unreasonable for me to go back and do this for every sample. And so I was reading a paper by a computer scientist at Stanford and saw how she was classifying pictures with a machine learning algorithm and thought, well, why can't we classify cells with a machine learning algorithm? And so I teamed up with Maria, and we developed Stellar, which is a deep learning method that takes this tissue graph and takes the molecular features about the cells and also the spatial features of the cell, and then uses that as training data, and then is able to annotate the cell type labels basically on a new data set. And another unique feature is that it's able to recognize novel cell types not present within the original data set. So if you look at one example of our imaged healthy human intestine, you can see that here we've mapped back the cell types that we've clustered back onto the tissue XY coordinates. And if we leave out this tissue and train on the other tissues and then try and predict with our algorithm, we can see that we get a high concordance, meaning that we have a high trust, or our trust is good within the Stellar algorithm. So we use this to annotate all of our samples across all of our donors. And we're able to start to see, going from the small intestine to the colon, we're able to see changes in the immune, stromal, and epithelial cell populations. And so I'm not going to mention everything here, but I will just say that we do see differences within immune cell population. Particularly interesting to me are this difference between the CD8s. In the small intestine, we have much higher abundance of CD8 T cells as compared to the colon. And that in of itself is interesting. But I was interested particularly in this variance that I saw within donors in the small intestine. And so we had some metadata associated with our donors. And one of those was people who had a history of hypertension. And what was interesting is that this variance was explained by people who had a history of hypertension. So people who had a history of hypertension had much fewer CD8 T cells within the small intestine as compared to those who didn't. And we confirmed that also this was true for endothelial cells, which you would expect to be the case for people who have hypertension. So this is something that's interesting, has implications for therapies, cancer, and can be followed up in the future. Another piece of metadata that we had associated with our donors was the body mass index. And what we saw correlated very strongly with the body mass index of individuals were this M1 macrophage population, which is an inflammatory macrophage type. It's an immune cell type in the intestine. And so as you increase the body mass index, you have this inflammatory macrophage increase. And none of our donors had a history of gastrointestinal disease, but it's also known that with an increase in body mass index, there's an increased risk for gastrointestinal disease. So potentially this is an early indicator or onset of such a disease. And because we had the spatial location of these cells, we looked at where they were located within the intestine. And Interestingly enough, the M1 macrophage, which is this one here on the left, is primarily within the mucosa, whereas the M2 macrophages were found across the intestine. And so the restriction of a macrophage subset was really important and really interesting. And so we looked at this, we followed this back up with a collaborator that I worked with, Magda, who was really interested in human macrophages. And she uses single cell RNA sequencing to further define further subsets beyond just M1 and M2. She's looking at 7 or 8 macrophage subsets. And we developed some markers for these 7 or 8 subset of macrophages and looked at where these were within the healthy human intestine, within colon cancer, normal breast, and breast cancer. And indeed, we saw further separations of these macrophage subsets, further indicating that these phenotypes, these subphenotypes, are also related to their functional and spatial restrictions. So, I mentioned going from this to this module. And so this is really what motivates this next computational analysis that I'm going to share. But I also mentioned, we can think about multi-cell modules, but we can also think about how these multi-cell modules form larger modules, right? How they fit together. And I think that that's also important to think about. So, one of the ways that we computationally do this is once we have the XY coordinates and the cell types, we take windows across the tissue, we measure the composition of cell types within those windows. So, maybe it's mostly T and B cells if it's a follicle, or epithelial cells if it's close to the epithelial. And then we take these vectors. So instead of a protein, we're now thinking about it as cell type composition. We cluster those into similarly composed cellular neighborhoods. And this describes the cellular organization across the tissue. And so what does that look like for the intestine? So here we have the fluorescence data. Here we've mapped on the cell types for this area from the Codex imaging. And then when we run the cellular neighborhood analysis, we can start to see and pull out in an unsupervised way the cellular neighborhoods or structures within the tissue. You can see that it separates the follicle into 2 structures, the inner and outer follicle. You can start to see the vasculature in yellow throughout here, the stromal features within gray. And we can look at this quantitatively as well when we define these neighborhoods based on cell types that are either the cell types that are enriched or depleted within that neighborhood. So for example, this plasma cell enriched neighborhood is enriched for plasma cells, but it's also enriched for other immune cells like CD4 T cells, dendritic cells, but depleted in things like smooth muscle cells or epithelial cells. And then we can use this, once we've defined all these neighborhoods, we can transfer these to our tissues. And not only are we comparing now cell types across these different locations, but we're comparing these structures across locations. And I mentioned these are the multi-cell modules, right? But then how do you think about multi-cell modules fitting together? And one example I'll give is the vasculature neighborhood that's found throughout the tissue. The vasculature here within the stromal area is probably different from the vasculature next to the follicle, which is different than the vasculature within the mucosa. And so how do we think about this? And, I think that we can learn from, other spatial analyses. We can think about, geographically, multiple neighborhoods make up a county, right? And then we can think about multiple counties then making up a state, and many states making up a country. And so it's this hierarchical spatial analysis that we already do within geography that we can then think about applying to the cells. And so these are example maps of the cell types for the small intestine and colon. We did the neighborhood analysis, just like I said, but now we're moving to the next hierarchy because we want to understand a broader spatial domain. And so what we do is look at neighborhoods of neighborhoods, or something that we call communities. And these are broader spatial events, and we can go even a step further and go up to things like tissue units. And the nice thing is that we can connect these quantitatively to each other. So we can look at the relationships of what cell types are enriched in which neighborhoods and which neighborhoods are enriched in which communities. And this can tell us about cell-cell interactions. It can tell us about multi-cell module interactions, which is really informative to the biology that's going on. And I'm only going to share one example today because of time, but you really can learn a lot from these type of graphs, even though it's a little complex in terms of the presentation. So one example is this adaptive immune enriched community. It is the intersection of multiple epithelial and immune cell communities or neighborhoods. And really interesting, if you look at it where it is on the map, it's this orange neighborhood or community, and it's at the bottom of the mucosa. And so here's the top of the lumen. And one of the interesting features of the bottom of the mucosa is that this is a stem cell niche for the intestine. And this is a really important and highly curated environment. And what we found is that there's also an enrichment for adaptive immune cells like CD4 T cells and CD8 T cells down at this area that wasn't previously appreciated before. The other thing that you can appreciate from just looking at this image, right, this is just a qualitative assessment, is that there's a layering of cellular communities as you move up the intestine. And this was known that epithelial cells differentiate as they move up closer to the lumen. But what we found was also that the immune cell composition changes as you move up through the mucosa towards the lumen. And now we were able to quantify this and see how exactly this immune cell composition is changing as you get closer to the lumen. And then finally, we also collected single cell RNA-seq and single cell ATAC-seq alongside these data sets. And we thought, how can we integrate this molecular features into our data sets? And so a talented PhD student in the lab developed this algorithm for connecting these data sets. And so now we co-embed these two single cell data set modalities into the same low dimensional space. We can see that their cell types overlap, meaning that there's a good integration there. And we can look at one example cell type like goblet cells and see that they both express MUC2, both in the RNA and protein, which means that there's a high quality of this integration. And so we thought, what can we do with this integrated data? And so we looked at these different communities that we've identified with the spatial data. And then we took the RNA data, the deep molecular data, and compared the expression between these different communities. And just to give one example, we looked within this neighborhood, the outer follicle neighborhood, we looked at the top differentially expressed genes in this neighborhood as compared to the other ones. And we can see a lot of different pathways we might expect to be present within the follicle structure of the intestine. So I think this is the tip of the iceberg, and there's a lot more things that we can understand from this type of data. So I'm just going to conclude this part of the talk, just say that, understanding clinical metadata associated with our the data that we collect is really important. It explained a lot of the variants that we saw within CD8 T cells or macrophages. I think, this macrophage subsetting and spatial subsetting, I think we'll continue to do with other cell types. And the more that we understand that, we'll understand some things mechanistically. Having this hierarchical definition of the intestine really enabled us to connect cells to multi-cell modules, to larger communities, to understand what's driving organization. And then, integration of multimodalities, I think, is going to be an important part going forward, just like this example that I gave. Okay, so now that was looking at healthy tissue. Now we're going to transition into a cancerous tissue. So I think this is really important because I think that the spatial aspect presents a new opportunity, and one for potentially diagnostics. Already, pathology is used in diagnostics, but if we can think about being able to determine multi-cell modules like an immune follicle as predictive of response or non-response to a therapy, I think that will be very helpful. But not only that, can we think about developing therapies that might interrupt some of these multi-cell module communications or be able to start even create something like a tertiary lymphoid structure? So I've been a part of a project. It's a multi-PI project across the nation that basically is looking at how chronic inflammation-induced cancers arise. So there are a number of cancers: esophageal, colon, stomach, and lung. All share this pattern of chronic inflammation that induces a change of state to the epithelial cells that go from normal to metaplasia, dysplasia, and then finally to an aggressive cancer. And they all share this pattern where they have this chronic inflammation and it eventually leads to cancer. And we think of it as studying one shared pattern of disease, even though they're separate cancers. And what the data I'll show is the imaging that we've done for esophageal cancer, where we've looked at the normal tissue, metaplastic tissue, the dysplastic tissue, and the cancerous tissue. So, we did the codex imaging, the clustering, and these are the neighborhood maps from two of the example tissues. And this is normal, and this is a dysplastic. And what we can do then is look at the percent of, these neighborhoods along with normal, epithelial, metaplastic, dysplastic, and tumor, and start to see what cellular organizations align with the different diagnoses. But there was a high variance in these, and that's partially due to the actual biology. Each individual tumor can have multiple pathologies within it. And so what we did is we had a pathologist go through and grade all of the different pathologies within the samples. So for example, this one has 50% tumor, 40% normal, and 10% of this glandular epithelium. And this way, we can characterize the percent characterized by the pathologist versus the percent characterized by codex to see which neighborhoods correlate very strongly with the pathology. And we can do this for this healthy tissue and then also for metaplasia. And what we can see is that they correlate very strongly, which means that these multicellular modules, or the organization and composition of these cell types, are what's associated with the pathologist score. But what we saw is that multiple neighborhoods were correlating with the pathologist score, which meant that the overall condition was at a higher level than what we were measuring, so a higher spatial level. So we went on to develop these community maps like I described before. And here, I won't talk about all the findings, but one of the ones that correlated with the invasive cancer, one of the things that we measured was really interesting, which is this thing called Shannon's diversity. It's a principle from ecology, which basically is a measurement of the diversity of an ecosystem based on the percent and also representation of species within that population. And if we look at the normal, the community that correlated with normal healthy tissue, this was the least diverse, whereas this community that correlated with invasive cancer was the most diverse, which, this kind of it's actually really interesting where, the epithelial cells we saw de-differentiate into a metaplasia or more intestinal-like epithelial cell, and then to the, de-differentiate even further to cancer where they had lineage negative markers. But then also the stroma de-differentiated where it started to mimic more of an intestinal-like stroma or microenvironment, and then further de-differentiated where there was just increasing disorder or increasing diversity within the system. So to conclude this part, integration of this pathology or this top-down approach with these molecular or cellular diagnostics, I think is really important to further understand some of these unsupervised methods and data that we get. I didn't talk about this, but I think that there are plasma cells and fibroblasts from our data suggest that we can target those cells as well. They're not non-traditional cells. This structural de-differentiation that mirrors the epithelial de-differentiation is really interesting because it's a provocative idea. Maybe was it the stroma that changed before the epithelial cells and caused the epithelial cells to change, or the reverse? And I'm not going to talk more about that now. And then there's just conflicting forces, right? There's an increasing inflammation, but there's also increase in regulatory cells like Tregs in advanced tumors. Okay, finally, we went from healthy to cancer, and now we're going to a therapeutic setting where we're thinking about T-cell therapies. So cell therapies are not just for cancer, they're across the board. They're used for regeneration, for infectious diseases, for fighting autoimmunity, but a lot of the attention has been around CAR T cells for cancer, just because these are the first to really make a lot of headway and get approved. And reasons that people want to use cell therapies, you can program them, you can quantify them ex vivo, you can control them to some aspect. They're living, they can have specific targets, they can deliver payload. But at the same time, those benefits are also the downsides, right? A cell can traffic to a tissue you don't want it to. You can have cells take inputs from their environment that may make them not work as well. You can also have them deliver payloads you don't want and increase the toxicity. And so a lot of these things are spatially related, and trying to understand why cell therapies work or don't work is, I think, a really important problem going forward. So I studied this within a mouse model. So the previous two studies were within human data, and this is it with mouse data. And so what we used is this engineered TCR, which recognizes the melanoma antigen within mice. And so we give the mice melanoma on day 0, let those tumors grow fairly large by day 10. Then we take T cells, activate them, and throw them into the mice at day 10. And then we can take out the tumors from the mice over different time points and measure them with this Codex fluorescent imaging. And so these are example images over day 1, day 3, and day 5. You can see that there's increasing inflammation within the tumors. And you can see that by the MHC1 and PD-L1 expression on the tumors, but also increasing inflammation with CD45. And you can see a few cells pointed out of CD8 T cells infiltrating the tumor. And because we have Codex multiplex imaging, we can quantify all the individual cell types over time and how they're changing. But what was really important to me was, okay, what is changing about the T cells, and how is that impacting the environment and their environments? And so, thinking about this is, how do we quantify the spatial features of a cell? We often think about T cells and cells in general based on their molecular features. And we do a really good job of quantifying and characterizing and clustering or separating, memory versus effector, or central memory, or stem cell memory. But I thought, why can't we do the same thing with spatial neighbors? And so instead of the molecular features being what we characterize a cell on, what if we characterize based on their cellular neighbors? And so that's what I did for the T cells. So I created this T cell UMAP. So basically what this means is that each individual dot on this map represents an individual T cell. And each relationship, or the spatial relationship on this map, one to another, represents how similar a T cell's microenvironment is to another T cell's microenvironment. So these ones are maybe more similar. They're closer on the UMAP as compared to this T cell that's in this microenvironment that's very different. So we ran this for the data, and I'm plotting now the T cells found from day 1. And we can see that they're mostly aligned with tumor cells that are either proliferating or marker negative. Whereas by day 3, the T cells are getting into the space with more immune cells like dendritic cells. And by day 5, they're found across the board, and particularly arranged with inflamed tumor cells and macrophages. So, this is really interesting and shows the rearrangement of the T cell microenvironment within the tumor. And we can quantify that through unsupervised clustering, just like we would cluster cell types or cell subtypes, and quantify how the T cell microenvironments change over time. And this way, we're able to characterize the T cell microenvironments in the processes it goes in within the tumor. Another thing is that, my kids love superheroes, and people within the T cell field really love this idea of super T cells. And there are many different ways to make them. I chose one that is basically a metabolic inhibitor that, if you add this inhibitor, it pushes the T cells to a more stem-like state, and you get better tumor responses. I did, cytoff on these cells and looked at that. I got a similar phenotype that was described in this original paper, and then used these two, compared these two, actually these 3 different therapies within Codex. And, a lot of the data aside, what this showed, if we did a random forest modeling to show which cell types were most important in separating these different samples, was, interestingly enough, there were 2 tumor populations in CD8 T cells. CD8 T cells you might expect. But what was interesting is, if you plot the percentage of these inflamed tumor cells, which are these PD-L1 tumor cells, versus proliferating tumor cells, you can see there's an inverse correlation. And to me, this suggests a phenotype conversion from a proliferative state to an inflamed state. And alternatively, if you plot the percentage of CD8 T cells versus inflamed cells, you see a positive correlation, implicating CD8 T cells must be involved in some way. So, I started off with this multi-hierarchical, and Codex multiplex imaging helps us deconstruct tissues in this way, right? We can make 50+ molecular measurements spatially, that allows us to define cell types. Then we can use that to look at cellular neighborhoods and beyond to network interactions. But I would argue that, we should be deconstructing tissues, but we should also be reconstructing. And there's in vitro methods we can think about doing this, or tissue engineering, but we can also think about doing this computationally. And this is what I did. I built a multi-scale model, which basically you make each individual cell as an agent, which is a computational object with rules and properties. You allow it to work with other computational objects in a spatial environment, and then you see this emergent behavior from the simulations that you do. So I teamed up with Uran Agmon at Stanford, who built this software for modeling bacteria colonies, but now we have applied it to mammalian cells, in particular tumor and T cells. And if we look at this model, we can do simulations. Each individual point represents an individual cell or computational object interacting in this environment. And what we can see is that in a tumor growing, you can see the tumor growing over a 3-day period. We can then add T cells into this environment. You can see them secreting interferon gamma, causing changes in killing and death within the tumor. We can change the phenotype of these T cells and see how it changes their overall activity within the tumor. And because this is a computational model, we can then quantify what's happening over time. You can see that, obviously we can mimic these responses of delayed tumor growth by just changing the phenotype of the cells within the model. What's interesting, though, is that it's not due to killing. Usually people are thinking that CD8 T cell control of tumors is due to increased killing. Really, we're only seeing about a difference of 100 increased death, whereas we see a difference in 2,000 tumor cells by the end of the simulation. And so what this has resulted from is basically you see this zone. These little green dots are the T cells. These blue dots are these inflamed tumor cells, and the red dots are proliferating tumor cells. And what the better T cell therapy does is basically converts the proliferating tumor cells into an inflamed tumor cells and stops their proliferation and enables the T cells to catch up with tumor growth. And this is actually paramount to the T cells being able to control tumor growth in the long run. So it's really the theory experiment loop with data input from this imaging cytometry to our multi-scale models that really helped us figure out this mechanism of T cell tumor phenotype conversion along with the killing. So I'm just going to conclude this part. I think, these are static snapshots, and, we can take multiple snapshots, but I think infusing time by either doing that or modeling or some amount of computational inference is going to be important in studies in the future. For T cell therapies, I think that in addition to killing, we should think about these anti-proliferative programs when we're designing T cells. And, thinking about cellular therapies with the ability to change their own microenvironments and control their own microenvironments, I think is going to be an important parameter to continue to study in the future. And then, I didn't get into this, but one of the things that also separates the two T cell therapies is the less effective T cell therapies recruit additional immunoregulatory cells. And there's only a small fraction of cells within that population that actually do this. And so a small population of these inhibitory cells can be detrimental. So I'm just going to conclude now with some overall conclusions. I hope I showed that, we can use these imaging methods to describe individual cellular and quantify the order of a system. And this is actually important to the organization and understanding cellular mechanisms. I think we still have a long ways to go in terms of how we think about what cells are driving this organization. And this is especially important since, the next, vignette that I shared, disorder is bad. It's for this cancer, we saw increasing diversity. And so, but this kind of begs the question, can we reverse this disorder, right? Can we restore the tissue to homeostasis? And we should try and learn more about these mechanisms. And we can do that by perturbing a system where, that was the third vignette of these T cell therapies, where we can transfer these T cell therapies into a tumor and start to understand what about the phenotype of the T cells allows it to control its environment or impact its environment. But these are largely correlative studies, and I think that we need to start thinking about what things are we going to measure about a cell that allow it to change its microenvironment. And I shared this at the beginning of describing who I was and kind of setting up, it's important what we measure. And one of the things that has defined my time in my postdoc and a lot of this work is my own multicellular neighborhood. I'm surrounded by plenty of people that drive the human biology and provide the samples and provide the input of what's important clinically. I'm also surrounded by people who develop tech and apply that tech and work with me to develop new methods and measurements. And then, I'm also blessed to be around and collaborate with people who develop computational methods that allow us to understand this data more fully. And with that, I just like to thank, the larger group of people that I didn't put up on the slide before that help enable this work. My funding through the NIH and the American Cancer Society, and my family. And with that, I'd love to take questions if we have time.
Super cool. Thank you, John. Just a reminder, if anybody in the audience wants to ask questions, we've got microphones here on the side and you're welcome to line up. And then anybody who has questions online, you can put them in the chat and we will make sure that they get answered.
Is this working? Okay. As always, I really enjoy your talks. I have two questions. The first one is about your first topic you discussed. I was just curious to know if the selection of the neighborhood windows, is that arbitrary or is there kind of like a rationale behind the selection that you're comparing?
Yeah, great. So in terms of the size of the windows, meaning the number of nearest neighbors of the cell?
Or just areas?
Yeah, so there's a couple of parameters that you can change in that type of analysis. One is the number of nearest neighbors that you select that go into the vectors. And this is basically defined based on your biological question. Is your question, do you want to describe structures that are within, smaller distances, 20, 30 microns, the nearest neighbors of a cell, or do you want to describe larger tissue features? And so that's one thing to take into account is what's the biological question. What I wanted to do with this first study was look at these smaller microanatomical units like the vasculature or the inner or outer follicle. But then I knew I wanted to layer on this additional, layer that's a higher order that used the subset. And so that kind of drove how I chose that feature. In terms of the number, I think that this also relates back to the biology. If you're more interested in broad categories versus, very distinct, it's kind of like when you do single cell clustering for either a high parameter flow or CyTOF or single cell RNA sequencing, you can always continue to subcluster, right? And it just depends on the biological question. Does it make sense to further subcluster? Yeah.
Okay. And my second question is about your third topic. In your workflow, you treated them with CAR T for a day 10 or 11, and then you allowed that to go for the next 2 or 3 days. I was just curious to know if you did any long-term studies to see how the neighborhoods or communities might change after long-term exposure to CAR T.
Yeah, great question. Yeah, so some of the tumors, they grow very quickly. So it's harder to go much beyond that, but we're doing a study right now to look at maybe 10 days. I think that would be the max just because the ineffective T cell therapies, the tumors grow out and they're too big for the mice. But yeah, I think that potentially also with a different tumor model that allows a longer term growth, that would be interesting. Yeah.
Thank you.
Yeah.
Thank you very much. I also really enjoyed your talk. I was wondering if you can share a bit more on the staining protocol for the images, what type of microscope you're using, and how do you work with 60 fluorophores or 120, as you said, or potentially without spillover and
yeah. So technically, you can use any microscope. I've built I with another electrical engineer built our own microscope, and use that. We have a Keonce associated with much of the data I showed was basically using a Keonce microscope that was associated with the microfluidics device that handles all the fluidics. The way that we avoid the spillover is that we do it in cycles. So at any one point, we're only adding 3 complementary sequences of DNA barcodes to the antibodies, which also have a DNA sequence. So for example, in cycle 1, we'll image CD3, CD4, and CD45. We'll strip off those after we image them, and then we'll throw on CD20, cytokeratin, and FOXP3. And then we just continue to do that cycling until we've imaged all of them. So we don't have to worry about the fluorescent spillover because we're only using 4 channels at a time. Yeah.
Okay, that makes sense. Thank you.
Yeah.
Really beautiful talk. Really enjoyed that. But I have a question about, at the very beginning, we were talking about I think you were manually encircling each cell type, right? So that you could quantify their nearest neighbors, et cetera. And you said that that was not a perfect solution because you had, errors along the way. And then I think you employed some computational means of doing it. How do you know? I mean, everything you do, which is really beautiful visually, is all dependent, it seems to me, on how well that initial step is. How do you, what kind of error detection method do you have to make sure that that stage of your process is working as well as you think it is? Because I can imagine a lot of this is, these beautiful images come out. There's some confirmation bias that could be there that you might not think about, and everything depends on how well you do that. So I'm just curious how you make sure that your error rate is really low.
Totally. Yeah. So great question. Initially, we were using methods like it's computational technique to separate out the cells, like a watershed segmentation. We moved to a deep learning method where we're able to now basically throw in the images and it annotates based on training data. And that works much better. Still not perfect, like you said. And that's why it takes a long time in the cell type annotation, is it requires a person in the middle just to make sure that what we call a CD4 T cell is a CD4 T cell. Because you can get bleed through of CD4 that are very close to B cells, right? And you might get bleed through the signal a little bit. Overall, it's pretty accurate, I would say. It's not perfect, so there are people working on making it better. But the place where the quality control comes in is at the clustering step, and you basically just look at the data and make sure that what you are annotating is correct.
Yeah.
And it's not going to be perfect, but
Right.
The 98%, I would say 95 to 98% correct.
Yeah, and so then if you see something novel, for example, how do you kind of work back to make sure that it's really novel and not some error in the way that the clustering occurs?
Sure, yeah, especially if it's something like a new cell type, that's something that you go directly back to the images, and it's just like evaluating a traditional IHC where you don't quantify, like you overlay, you make sure that the cell really is triple positive, for example. But so definitely multiple steps of confirmation. Yeah.
Thank you.
Yeah.
All right. Thank you. Great talk. Thank you for giving us your time. This is really appreciated. So I did have a question a little bit. Obviously, you're out of Gary's group, and you're using the little bit more higher res system, but there's also imaging mass cytometry. And I'm wondering how, if perhaps the imaging mass cytometry itself wasn't resolved enough for these studies, if it was, and how that compares in your choice to use the Codex versus IMC.
Yeah, I think that there are a lot of different systems. There's IMC, there's cyclic IF, there's many different techniques. each comes with its own advantages and disadvantages. For example, within the lab, we also have this method where we attach these metals and then stain. It's mass cytometry, basically imaging mass cytometry. And that's really great because you don't have tissue autofluorescence to the same extent that you do in this Codex multiplex imaging. One of the things is just the ability to run them quickly and a lot of samples. That machine is a little more expensive. And so we have fewer of those, well, only one in the lab. And so, yeah, I think the potential for barcoding of antibodies is also bigger in the oligonucleotide space as opposed to the metal space, because there you're limited based on the isotopes. Not that that was so much of an issue with these studies, because we were usually around the 50 to 60 range anyways. But yeah, I mean, I think many of these techniques are equally as good. You know, they achieve single cell resolution.
And kind of one question that kind of jumped into my mind, and this may be even a little silly, but so we spend in our cell segmentation, we spend so much time trying to differentiate the cells and try to eliminate those little carryovers. But have you done anything to actually look at maybe scoring those direct interactions? Because those are two cells together, two different cells you're imaging, but we just see them in the same area space due to the sectioning. I mean, have you thought about potentially using that in your neighborhood and your community analysis as well?
Yeah, so I guess just so I understand the question, is it that you're saying do we look at cell-cell interactions, or do we try and use this error rate of the spillover to quantify interaction?
More like the error rate of, so when you're labeling tissues, you'll sometimes get, because the cells aren't perfectly aligned, you have two cells that almost are inheriting the same space because they were, it's you're looking at a two-dimensional, three-dimensional tissue.
Yeah.
And I was like wondering if maybe that overlap that we try to eliminate and kind of segment around would be used in some kind of way to affirm your nearest neighbor and maybe even score your community.
Yeah, that's interesting thought. I think that traditionally we've been looking at just cell-cell interactions based on distances and distance permutations, or like a neighbor and neighbor permutation type testing. But yeah, that could be really interesting in terms of an approach. Yeah.
It's just one of those times when you think about it, it's like you spend all this time to get rid of things. And then it's like, well, would that be interesting?
Totally. Yeah, thank you.
I'm gonna ask just one from online as well. So fantastic talk. Are you planning to include the microbiome as another factor influencing the way the structures form and work, which is a question that I have as
well. Great question. Yeah, I think, obviously we know that the microbiome is really important. I think for these human samples, these were from deceased donors, and so they clear the gut right away. And so that is not, we weren't able to do that. But, in the future, especially with studies with mice, that would be really cool to include, definitely. We have the multiplexing space. And so, yeah, that would be really interesting.
Thank you again, John. That awesome talk.
Oh, okay, the title change is already up there. It's my pleasure to introduce Dan Freeman from TerraFlow. Since first teaching himself how to code when he should have been studying for the MCAT, he says he's been obsessed with using science and technology to hack our understanding of challenges in health industry and society. Dan first learned about flow cytometry when he was working with Prateep. And everybody here in FlowTex knows Prateep very well when he was at New York University Medical School. When his lab shut down during the pandemic, he turned his attention to flow analysis. He founded TerraFlow in his Brooklyn walk-up, and eventually helping clients from Novartis and the Cleveland Clinic extract rich biological narratives from their single-cell data sets. He continues to grow TerraFlow while completing his PhD in computation biology at Harvard. and as well as teaching the undergraduate course, Moonshots, Ideas and Technology Shaping the Future of Healthcare. I crossed paths with Dan at Cyto last year when we were in Dave Gravano and Laura Ferrer's workshop. You're going to hear about that in a little later this morning. He's a delightful individual and is more than happy to introduce him. And the title of his talk is Flow Analysis Must Become Interpretable. Dan.
Imagine you carry a child for 9 months, and the moment that child is born, somebody takes it away and says, I'll come back in 16 years and tell you how they did. Imagine how angry that would make you. When I worked in flow cytometry, my projects were my babies. I was the one who knew the biology, who designed the panel, who ran the experiment. And at the end of the day, I wanted to be the one that comes back to a collaborator with a story to tell about their data. But if good science is about storytelling, then so many of our bioinformatic tools take us in the complete opposite direction. We grasp at UMAP plots or heat maps, trying to piece together something significant, or we just outsource the analysis to a bioinformatics core, giving up our babies at their most precious moment. My name is Dan Freeman. I was a founding member of the Precision Immunology Center at NYU Medical School. I study artificial intelligence at Harvard University, and I'm also the CEO and founder of TerraFlow, together with my mentor, now co-founder, Dr. Prateep Chattopadhyay. Today, I'm going to tell you about a method I developed that can take any single cell data set and explain it back to you in plain English. I'm going to show you how we use this method to do a major study of Hodgkin's lymphoma in-house with a core of 4 people. And I'm also going to show you a platform we developed to make this tool available to everyone. Who can tell me what is the most common way to analyze single cell data today? You can just shout it out. But gating, we'll talk about gating. What about algorithms? What's the most common algorithm? UMAP, Disney, FlowSome. This is what a flow analysis looks like today. We partition our cells into 10 or so clusters. We check if any of those clusters are associated with an outcome, say drug responder, non-responder. And if we're lucky, we get to say, okay, cluster 3 predicts outcome with P less than 0.05, and it's got a little more of this protein and a little less of that protein, right? But if I'm a clinician in Houston, what do I do with that information? Right, the only way I can measure cluster 3 in my patient is if I run your 30 antibody panel, download your machine learning model, batch normalize our data sets. And at the end of the day, I couldn't even tell you what this cluster really is. What Prateep and I wanted to do is to develop a method that would allow scientists to mine the full depth of their single cell data while still returning results in a format that was meaningful to immunologists. Doing this meant returning to some of the most basic principles in immunology. We knew that immunologists define cell types by the combinations of proteins that they express on their surface. So we wanted to take this idea of combinatorial gating and apply it on a much bigger scale. The first step is to define a threshold for each marker. So if the data is bimodal like CD4, we put a threshold down the middle and define a positive gate and a negative gate. If the data is unimodal like PD1, we put a threshold on the right shoulder, and again, we define a positive gate and a negative gate. And then we start combining these gates combinatorially. So we say, okay, in patient 1, what percent of cells express CD3, CD4, CD3, CD4, CD95, et cetera, until we've enumerated every possible combination of 1 to 5 markers. So that gets us to this matrix where each row is a patient and each column is the percent of cells expressing that protein combination. Now, of course, through, say, CD4, CD3, CD3, CD95, exactly as if we were gating these phenotypes one at a time in FlowJo. We'll see it in a minute.
A little closer.
Excellent. Thank you. Oh, you have to. You do the clicker again? Excellent. Thank you. Combinations of 1, combinations of 2, combinations of 3. And you take this path through the network, it's almost like you're gating these combinations one at a time in FlowJo, right? The next thing we do is we exclude anything that is exceedingly rare. So as these combinations become more complex, they also become more infrequent. And so we set a detection limit at about 50 cells per sample on average. Then what we do is we measure how well each of these phenotypes correlate to our clinical label, and we project that information onto the network. So in this example, you can see CD95 GITR CD8 is very strongly associated with disease. It's very bright. But you can also see how that effect size kind of spills over into related phenotypes that have 1 or 2 markers in common. And so rather than just reporting all of these phenotypes as significant, what TerraFlow will do is go through the network and pick out the peaks and valleys to represent unique differences between our patient groups. So what this effectively allows us to do is take that really long, those thousands of disease-associated cell types, and reduce it to just a handful of unique cell types that each represent a unique difference between our two patient groups. That was pretty theoretical, so let me bring it back with a real example. We know that patients with Hodgkin's lymphoma demonstrate systemic immune dysregulation. If you give these expose these patients to an antigen subdermally, their immune response is not the same as a normal individual. But the exact cell types that drive that dysregulation are yet unknown. So what we did at NYU is we collected blood from 47 patients undergoing treatment at our hospital, as well as 23 healthy individuals. And then once we established which cell types were important, we validated our findings in a prospective cohort of 10 healthy individuals and 10 individuals with Hodgkin's lymphoma. The first thing we tried was a popular clustering algorithm called FlowSome. Now, we see that in FlowSome, only 2 clusters correlate to disease, and of those, only one of them, MetaClust3, validates to new data. The other piece we see is that these clusters are somewhat difficult to describe. So for example, I can see that MetaClust3 is associated, I see that MetaClust3 has elevated levels of HLA-DR, but it's not clear to me if HLA-DR is sufficient to capture that population, or if you actually need 2, 3, 4, maybe all 30 proteins to really recapitulate this phenotype. The next thing we tried was a deep learning model called Cell-CNN that David actually referenced yesterday. So now you can see our populations have a stronger association with disease, but they're only 2 in number, and they're still pretty difficult to describe. Here's that same analysis using TerraFlow. TerraFlow consistently identifies 8 or more cell types that are strongly associated with disease and that each validate to new data. TerraFlow also annotates every population with the smallest set of markers needed to gate that target population. So at the end of the day, you can say, here's a T cell, it expresses GITR and PD-1, and it separates our patients with a high degree of accuracy. As part of the study, we also wanted to know if the immune system normalizes after treatment or if the disease leaves some kind of permanent mark on the immune system. And it turns out that it does. Patients that had Hodgkin's lymphoma and then recovered still demonstrated global signatures of T cell exhaustion, including combinations of markers like GITR, CD152, and CD272. This is the first time we've described specific cell types associated with Hodgkin survival, and we can start getting at how these cell types might impact future treatments like immunotherapy or COVID vaccination. Now, earlier I told you that we use thresholds to partition positive and negative gates. But the problem there is that setting thresholds can be a very time-consuming and subjective process. If I ask 3 cytometrists in this room to gate my PD-1 expression, I very well may end up with 3 very different answers. And if I employ a few automated gating algorithms, I'd probably just end up with 3 more. And so Prateep and I wanted to know, what if we could do this discovery without using gates at all? In traditional flow cytometry gating, okay, time for your imagination again. In traditional flow cytometry gating, we draw a rectangle, and every cell inside that rectangle gets a value of 1. Every cell outside that gate gets a value of 0. And the average of all those 1s and 0s equals some percent positive, say 8% double positive. So you would see here, all the 1s would be red, all the 0s would be blue. It would be a binary. With our non-gating approach, the brightest cells still have a value of 1, the dimmest cells still have a value of 0, but now we introduce a continuum of values between them. And what this allows us to do is to approximate that double positive population without relying on a single cutoff. So if this image were covered, you would see the top corner would be red, the bottom corner would be blue, but you would see a range of colors in between. We find that this non-gating approach can approximate classical gates. So what we did is we took that initial cohort of 70 patients and identified disease-associated cell types using our non-gating method, so no thresholds. And then what we did is we took our validation cohort of 20 patients and had a flow cytometrist hand gate those cell types using traditional gating the way a clinician might do with FlowJo. And it turns out that they validate. So in other words, you can easily translate back and forth between non-gating and classical gating phenotypes. The difference is that with our method, you can do the early discovery without having to rely on thresholds. So where are we now? We've done an exhaustive search for disease-associated cell types. We've validated our method using Boolean and non-gating methods. And we've also defined each cell type with the smallest set of markers needed to gate that population. But no algorithm can do everything. And at the end of the day, you still have to go into PubMed and figure out what these combinations actually mean. Actually, TerraFlow can help you with this too. We immunologists don't have time to read every single paper that comes out in their field. So we're starting to use ChatGPT-enabled technology to query thousands of biomedical journals and synthesize what people are telling you about your selected cell populations. So when the algorithm tells you that GITR PD-1 is important, we can tell you that that population has been associated with increased reduced effector function, increased regulatory activity, and the development of immune tolerance. You know, yesterday we heard Johnny speak about the importance of being a translator for clinical collaborators. Here's a platform that can take single cell data and explain it back to you in plain English. And it's doing it in a way that anybody can instantly understand and replicate on their own instrument. The best part about TerraFlow is that you don't have to be a bioinformatician to use it. All you do is upload your FCS files, label the patients you want to compare, and hit submit. In 24 hours, you get back a report that lists all of the immunophenotypes important to your disease. This report is basically a preprint about your data that tells you what cell types were important, how those cell types relate to each other, and how these phenotypes connect to current biomedical literature. And so you can imagine maybe you're a flow core manager who couldn't provide data analysis because you didn't have a full-time bioinformatician. Now you can using TerraFlow, and it's presented in a format that anyone can pick up and instantly understand. We've now piloted TerraFlow with our first wave of early adopters. And we've seen, one, that TerraFlow allows people to extract insights from their data that would have been ignored using traditional methods. But on a more important level, this technology allows flow core managers to step into the role of translator and come back to, could be a biologist, a clinician, a pharmaceutical executive. It doesn't matter because it's a format anyone can pick up and instantly understand. I'll close with this idea. Making flow cytometry analysis complicated is easy. Creating a neural network is easy. Creating a UMAP embedding, a flow sum clustering, that's easy. What is hard is the decades of firsthand experience it takes to really become good at flow cytometry. And I build these tools so that the people who understand the data and the biology better than anybody else in the world are the ones that come back to their collaborator with a story to tell. Thank you very much. Yeah, question over here.
It was a really nice talk. I like the way you presented.
Thank you.
My question is, okay, you say no gating, you start the data with the mapping and everything. So how do you address doublets, Z cells, autofluorescence, and all those things?
Yeah, thank you for that. So right now, we are working towards that. Right now, we usually start with a general population. So for example, in this study, we gated out live CD3 positive T cells because that's the population we were interested in, and we started our analysis there. But we're also interested in adding additional layers that will allow us to truly start from data fresh off the instrument. Thank you for that.
I just had a question kind of echoing that a little bit of, is there a way that you can include rare populations? Because in some labs that's kind of their focus is to look at how rare populations influence the whole network. And one of your things is to exclude those.
Yes. So we set the detection limit at 50 cells per sample on average. That means that you could have, let's say you have two cohorts. One cohort could have 100 cells per sample. The other could have 0 cells per sample, as long as the average is about 50. So we will pick it up in those instances. The reason we set the threshold, I mean, you can set the threshold wherever you want, but the reason we do this is because as these populations get more complex, as they become more rare, they also become more noisy. And so these really rare populations, often they don't validate. At least with this method, they don't validate when we go into a new cohort. Yeah, sure.
Super cool talk.
Thank you.
And great work. I'm excited. I don't know if you have a paper out, but I'd love to see it at some point. The question that I had is with this exponential amount of combinations, how do you think about the multiple hypothesis testing, and how do you think about correcting that, especially since dependent on the study, you'll have a different number of markers and also a different number of cell types that fall into that category, right?
Yes, this is a huge focus of our research. I will say this, we evaluate about 100,000 combinations per data set, but our algorithm is biased towards selecting the fewest numbers of markers possible. We kind of rein that into 1, 2, only going up to 3 markers if necessary to define that population. That helps. Now, to check our work, we have a few things. One, we do a FDR adjustment. We do cross-validation within the discovery set just to make sure our overall method is valid. And in this paper, we did a prospective cohort. So we actually assayed these patients after our data analysis was complete and verified that our selected phenotypes validated the new data. Yeah, thank you for that.
I want to quick get in. We do have some online questions as well, so
Okay.
we're going to kind of do.
Okay, it was a short talk, so let me hear it. Yeah.
So we do have one concern about one concern from online about ChatGPT being ignorant of research prior 2021, as well as potentially giving misleading interpretations of immunological knowledge.
Yeah. Thank you for that question. ChatGPT is wrong sometimes. And the other part is that ChatGPT always thinks it's right? We use it as a starting point, so that before you even open PubMed, you have some idea of what this population looks like and why it might be important to disease. And that's the way I use ChatGPT when I was writing this paper.
Thank you.
Question.
Hi, Dan. How you doing? So a couple of things. One is, what is the minimum number of markers you would anticipate necessary to upload into the system to give out phenotypes that would be meaningful? That's my first question.
One
One marker.
If you want to do one marker, you can.
Okay. What was my second one? If you were to upload something like, so you're using the example of non-Hodgkin's lymphoma. Let's talk about a large granular lymphoma. If you upload that into that, have you tested the training set to see if the same phenotypes pop out? Similar phenotype cancer, other than, size, CD10, things like that. Do you get the same sort of populations of interest that pop out, or is it completely different?
Are you saying the same population in Hodgkin's as we do
Similar
in
phenotypes as Hodgkin's, yeah.
we don't have access to that data, but if you do and you want to collaborate, I would be happy to see it.
Issues would be, of course, like the marker subsets, right? So that's again,
Yes.
We would identify it using a different set of markers
Yes.
at University of Connecticut versus what NYU
Yes.
would do.
Yeah, the analysis starts from fresh every time you upload a data set.
Okay.
Yeah, that's right. And I just want to say one thing about the ChatGPT thing you mentioned. The thing about flow analysis is that you don't know what you don't know. And sometimes what tools like ChatGPT can do is they can turn us on to a mechanism that we maybe never considered before. So it can be a source of creativity in your analysis as well.
Oh, I appreciate that. I also kind of think of it as how you deal with people in general. There's a lot of times you'll be interacting with someone and you have to actually wonder about their level of expertise and where their answers are coming from. Because having been a father, I understand that sometimes you have to just give an answer. It doesn't have to be right. You have to give an answer.
Yes.
So, so I do have a couple general questions from online that are really interested, and I'll kind of cluster them. So one in particular is the analysis pipeline takes 24 hours, and then also followed up by, if doing this, do you have to go ahead right as of right now, like in FlowJo, gate on the CD3 population, and then have that sent to the algorithm?
We've done both. We've done studies where we just took fresh compensated data and used that as a starting point. In this case, we knew a priori that we were interested in T cells. So we gated that population first, kind of narrow the scope of our analysis. And then we put that into the TerraFlow. But you can start now with raw data, and we're working on improving that as well.
And then the other question was the 24-hour period. Is it necessary? Is that kind of a server thing? Is that just the algorithm takes that length of period of time?
It's up to 24 hours. It can take less.
Thank you.
Yeah, question.
Then I'm just wondering how you deal with the batch effect. We're approaching this.
Yeah. We don't have a built-in feature that deals with batch effect yet, but this is something we're very interested in. We also have early results, very early results, suggesting that our non-gating approach may help mitigate those batch effects because you're no longer relying on just one cutoff to define positive and negative events. So that extra flexibility may help. Yeah, be determined. Okay, thank you very much.
Great.
Welcome back from another wonderful coffee break for those of you in person and for those of you online. I hope you grabbed a nice refreshment as well. It is my privilege to introduce Meredith Weglarz, who is currently managing the flow cytometry core at the University of Texas Medical Branch in Galveston. She previously helped manage the Stanford Shared Facts facility before moving to Texas last year. She has worked in cores in both academic and industry settings and is a certified specialist in cytometry with over 10 years of flow cytometry experience and knowledge. She's an active member of ISAC and is a community member or committee member for the Flow Content Committee. And she is a new member to FlowTex, and we are so appreciative of all the work she's done already this year. We are excited to hear her talk. I'll just use what's in the box.
Let me just hide this. All right, so as Amber said, I'm going to talk to you about being in a flow core and the most common phrase of
Well, I just put my panel together with whatever was in the fridge or in the box. So I'm sure we all have a lot of experience with that. And it's very, very common. So I have a really great example for you about why, if you just spend 5 minutes talking to your local flow cytometrist, you may get some better data than what was in the box. So I'm pretty sure we've all had this experience. A grad student walks into the flow core. The flow cytometrist says, "Hey, what are we doing today?" The grad student says, "I'm looking at marginal zone and follicular B cells. I made a panel with whatever was in the box. It'll work, right?" The grad student hands over the panel. This is the initial panel. Looks like they attempted to titrate it at some point. The flow cytometrist shakes their head and says, "Yes, but really this could be better, but let's see what the data looks like." So let's go ahead, let's take a look at what their data looks like from this panel. So again, this is a marginal B cell follicular zone marginal zone and follicular B cell panel. But they were also hoping to get some information about germinal center B cells as well. So the beginning of the getting is pretty standard, but the first thing that we noticed when we ran this panel is they actually had no marginal zone B cells. And this was infected mice, so they should have had marginal zone B cells. There was no reason for there to not be marginal zone B cells. The next thing that I noticed is that to confirm marginal zone B cells and follicular B cells, they need IgD as well as IgM to confirm, which is pretty standard, and we should have a nice population that looks like this if we have the proper markers to confirm them. The other thing that we noticed was there were no germinal center B cells. Again, this was a vaccinated infected mouse. There should have been germinal center B cells. So great, they just spent all this time. It was about 6 months of an infection, so probably around $20,000 experiment in the VSL3, and they got little to no information because they didn't spend a few minutes talking to us. So we sat down after their talk and we went over their panel. And we've heard a lot of panel design talks, so I will not overly bore you with the panel design. But the first thing that I normally do is talk to the researcher. What's your question? What's your hypothesis? What are you looking for? Can flow cytometry really help answer your question? Or are you doing flow cytometry when you should be doing imaging? Sometimes they overlap and sometimes people aren't really sure what the proper technique they should be using is. So we talk a lot about that. And then I have them start making a list of the antigens that they need to answer their question. So what antigens do you actually need to get the markers to confirm that they are marginal zone B cells or follicular B cells? And then I also have them, this is similar to your primary, tertiary, secondary antibodies. I generally have them rank them on level of importance because a lot of my users don't understand a primary, secondary, and a tertiary understanding. So if I just have them mark them very important or not important or mid-level, that generally gives me the information about primary, secondary, and tertiary. And then I also, because I'm not an expert, I'm not an immunologist, I have them indicate to me what the reason that they need that marker is, because I don't know every single population under the sun. So we started there and immediately we went ahead and added IgD because it wasn't originally there. Then we started to look at the antigen density. And again, we talked about this a lot yesterday and the day before, whether it's an on-off signal or a smear. So here are some examples of an on-off. So B220 CD3, where you clearly have a positive versus a negative, a positive versus a negative. I consider those on-offs. And then a smear population where you may have, in this case right here, it's not super clear what's positive and what's negative, though you really do have a positive and negative. So I have them start indicating that information for me if they know. A lot of times they don't know, so I end up doing the researching to figure that out. And then I also ask them if they know how highly expressed the antigen that they're looking at. We add all this information to the table. So we keep our antigens and we add expression level and whether it's a signal is on, off, or smear. From there, we start looking at co-expression. And I believe Anna talked about this. She likes them to draw a gating strategy. This is also my preferred way of getting the co-expression as a flow cytometrist. I'm a very visual person, so I like to see the gating strategy. So I generally have them draw a gating strategy. However, a lot of my users are not visual people. So they will go ahead and give me a list like negative, positive, this is what I expect. And I will help them, just like Anna mentioned, that she will go over the gating strategy with them. I will also help them come up with that gating strategy that they need. But basically, what cells are you expecting, positive and negative? Do you expect co-expression or not co-expression? So we go over that. And then we start talking about fluorochrome brightness. This is again something that we've heard a lot about already. So as you guys know, there's plenty of these fluorochrome brightness charts. Here's the one from BD. This one's from Thermo Fisher. This one's from Cytec. They're all a lot. They're generally overwhelming, but your flow cytometrists have a lot of this knowledge already piled up. And generally, we know this stuff pretty quickly. Oh yeah, this color is brighter than this color from experience. So feel free to use us to your advantage because this is wonderful information that we already have. So then I start matching those expression levels of those bright expression levels to a dim fluorochrome and vice versa. So I want everything to come out like somewhat of a moderate expression level. So I want a bright fluorochrome with a dim antigen so that I find somewhere in the middle. Moderate to moderate, that's going to stay in the middle. If it's a bright antigen, I want a dim fluorochrome. If I put bright on bright, I'm going to get screaming, and then I can't do anything because it's off the charts. For example, CD45, I don't know how many people in my flow core put it in PE. PE is bright. CD45 is bright. Let's move it. So that's the big thing. So I spent a lot of time going over this. A lot of my users also ask me, like, how do I break up one of these guides? Just split it somewhere. I'm asking you for bright, moderate, and dim. Give me like a 1/3, a 1/3, and a 1/3. Like it's okay. It doesn't have to be perfect. You can make a best educated guess here, guys. And then I start actually choosing the antigens, and I want to make sure the antigens that are co-expressed, their fluorochromes are not too similar. So for example, if we look at IgM and IgD, this is going to be our confirmation markers for our follicular zone and our marginal zone B cells. And they're going to be positive and they're going to be all sorts of different expression levels. So we really need this to be well separated so that we can clearly see. So in this case, they were also both bright expression levels. So we want to match them with dim fluorochromes. So in this case, I just went ahead and I chose 2 fluorochromes. And here's your spectra view of those 2 fluorochromes. So I'm a huge fan of Brilliant Violet 510 because it's pretty dim. So it's great for some of your super bright markers. So the blue is Brilliant Violet 510. It's excited with the violet laser, and then you have an emission around 510. And then the other red one that I have here is Brilliant Violet 711. It is a little on the brighter side, but it matches very nicely, in my opinion, with 510. There is a small amount of overlap, but they are very different, even though they're both excited by the same laser. So in this case, I went ahead and chose those 2 colors. So when I'm thinking about my whole panel, this is my original panel here, and here's all the information that I collected. Again, this is very similar to what Anna was talking about yesterday. She has a giant Excel spreadsheet that she gets all that information and puts it into. So when I'm looking at it, the first thing that I'm going to do is make sure that we add IgD, since this was originally missing. PE was really bright, and IgM is a bright expression marker. So I want to go ahead and lower that fluorochrome down so it's not so bright. The CD23 and CD21-34, which are going to be our original markers for follicular zone and marginal zone and follicular B cells. So we went through and double checked that they were using the right clones because they didn't actually detect those populations in the beginning. And we decided that we would titrate them first to see if they still worked. If they worked with a titration, then we weren't going to change them. As this specific researcher, their lab does not have a lot of money, so they can't just change their entire panel just because it didn't work. So since these two markers they had a lot of, we decided to check the titrations, see if we could actually detect what they were looking for, and go from there. CD38 was in APC. CD38 is pretty bright, and APC was way too bright, so we needed to change this. GL7, which is a popular marker for germinal center B cells, is pretty moderate, and it's on Alexa 488. Again, it didn't work in their original panel, but we decided that if we looked at a positive tissue when we were titrating, such as a pyrus patch, which would be highly expressing for GL7, we would see what the proper titration was and if the antibody was working. If it was, we would continue forward with that antibody. CD138 is really, really dim, and they weren't seeing any plasma cells. Plasma cells was like a secondary question that they had. So we decided we need to change that antibody to get it brighter. And then finally, we have our B220 CD3 live dead. We would just figure it out at the end for those three if we needed to change them. So we went ahead and we started to change our antibodies. And earlier, we had mentioned that we picked Brilliant Violet 711 and Brilliant Violet 510 for IgM and IgG. So we went ahead and start filling that in. And, and then CD38 will look at with GL7 to look at our germinal centers, but it will also help us determine our follicular and our marginal zone. And APC was just way too bright. So we went ahead and changed that out for a more moderate fluorochrome. And we just went ahead and picked PerCP for that because this lab really loves PerCP Sci 5.5. It's like their favorite fluorochrome ever created. So if they could buy every antigen in PerCP Sci 5.5, it would make them very happy. So, you know, sometimes you just have to make the researcher happy, and that's okay. So, yep, we chose PerCP, and it was perfect. And then the problem then became that CD138 was already in PerCP, and this was looking at our plasma cells, and CD138 is pretty dim. But since we needed a bright one and we already got rid of APC, we decided to put APC in CD138, and that would give us a nice signal. And then we just went ahead and we changed our B220 CD3 and AmCyan. This is actually in the second version that we changed these fluorochromes. In our first version, again, the lab didn't want to spend a lot of money, so we didn't change them right away, but we ended up changing them in the second version. And that was only because the PI was like, wow, the data looks so good. Is this the best we can make it? So then we went through and we changed it. So we ended up making a dump gate and adding CD3 and live dead into a similar channel. And we gave B220 a slightly brighter fluorochrome because B cells were the of interest population in this case. So that kind of made it through how we designed our panel. And we ended up leaving PE, Brilliant Violet, and 605 open for future additions, which was great because a month later, obviously, they wanted to add more markers, and they were memory B cell markers, and they were all super dim. So we had PE, Brilliant Violet, and 605 open, and we were able to just add them right in for super easily. So that was great. The next thing that I had them do in this case was actually titrating the antibodies. And I know that we've heard a lot about titrating the antibodies. But in this case, the lab was really just using like 2 microliters of this and 1 microliter of this. There was no rhyme or reason. They're like, oh, we didn't get good staining last time, so let's just triple the antibody concentration. And I was like, what? What? So I had the grad student go through the process of titrating the antibodies, really for a learning experience and to understand how to make a good panel. So for those of you who don't know, titrating finds the ideal concentration for each antibody, and oversaturation or overstaining can lead to inconsistent results and increased backgrounds, and therefore it's not always great. And then understaining or undersaturation can also lead to the same kind of problems. So in this case, in my lab, I just have them run 5 different titrations. So 1 to 50, 1 to 100, 1 to 200, 1 to 400, 1 to 800. So pretty simple. And then I have them calculate the stain index, which I cheat a little. I just do the medians minus each other over 2 RSDs. And in this case, we found the ideal concentration was 1 to 400. And they were like, oh my God, I've been using so much more antibody than I have to. And I was like, this is why we do this process. So that was a lot of fun. And this grad student really learned a lot. And she really appreciated the whole titration process, even though she complained that it was very painful in the beginning. But now she's like, oh, it's so easy. Why doesn't everyone do this? I was like, do you want to come to my next talk? So anyway, so she titrated all of her antibodies, which was great. And for those of you who are like, I have no idea how to titrate, many flow cores have our titration protocols up online. I'm sure Anna and Rui also have their titration protocol online. I'm sure Joel has one. I'm sure Karen and David have one as well, pretty much so everyone. But if you're looking for one, the UTMB Flow Core also has one, so feel free to check us out on our website. I recently made it. It's very beautiful. Check it out. But most importantly, and I was talking to some of the other FlowTex committee members about this, please don't forget your unstained cells. Like, it's very easy. So just make sure you're adding another sample for unstained cells. I know that a lot of companies recommend doing more than 5 titrations, but generally you can get away with 5 titrations. It's not that hard. And if your samples are really messy, for example, liver, you might want to first titrate your live dead antibody, and then go ahead and add your live dead to your titrations. But if you're going to do that, don't forget that that may require some compensation. So please be nice to your flow core. So then our next step was to go ahead and test the panel. So we went ahead and ran the full panel with our titrations, and things that we are evaluated was whether or not our populations were clear, if we needed any FMO controls. So could we gate without FMO controls, or did we need them? Did any titrations need to be reevaluated, or so on and so forth? In this case, we got really lucky, but it's also only a 10 color panel. So it's a little bit easier than some of those 36 color panels that we were talking about earlier. Even when you're getting up to 18, generally, if you test a panel, it might not look perfect. But in this case, we got pretty lucky. And then your next step would be, again, we talked about redesigning the panel. In our case, we were lucky we did not, but sometimes redesign could be as simple as going back to your titrations and double checking them. It could be changing 1 or 2 antibodies. It could be adjusting your controls. Again, we got lucky. So I just wanted to show you a comparison of what the panels looked like. So you can see how high antibodies they were using prior to titration, and then post titration, they're using significantly less antibody per 100 microliter. Most of the fluorochromes did end up to be different, minus these 3 down here. But again, the PI was extremely happy with the results initially, so we went ahead and made the panel as best we could. And then some of the data comparison from the original. We now have germinal center B cells, which we didn't see at all at the first round of infection. And the user did end up showing me a bar graph. There was actually some differences between her 2 infections, which was great. So germinal B cells were present finally. Our marginal zone B cells showed a beautiful population. Even our follicular B cells showed a beautiful population. One thing that you'll also notice is the number of events. She was initially only recording 20,000 events, but these populations require more than 20,000 events to find them. So that also changed. And then again, the IgM and IgD just wasn't present before. And now we can clearly see that our marginal zones are high for IgM, low for IgD, whereas our follicular B cells kind of have a nice gradient in IgM and high IgD expression. So that was absolutely beautiful. And my biggest take home message, which I think pretty much so any flow core would tell you, is flow experiments are very expensive. And it's not just the flow antibodies or the flow time that it's expensive. It's the time that it takes you to get to the flow instrument. Don't waste your time and your money on a poorly designed experiment. We are here for a reason, so please talk to us because we will help you, we will save you money, and your science will be saved for it. A short talk can go a really long way. This panel was redesigned in about 15 minutes. It doesn't have to be a 30-minute to a 6-hour conversation. With some of the smaller experiments, those data can be improved. It doesn't have to be an 18+ color panel for you to talk to your flow people. So please use us as a resource. We are here for you. Even if you think that you have a flow expert in the lab, this lab did have a flow expert. So yes, that's my take home message. I think I talked really fast, but if you have any questions, I'm happy to take them. There's microphones down here, so if you want to come on down.
Thank you for the very interesting results. So my question is, were the data validated by imaging? Would you know about it? Because most especially the B cells were dependent on the localization in the lymph node. So it's most especially in the marginal zone
Yeah.
B cells, they are located afar from the follicle on the lymph node.
So this is a grad student and she is actively working on doing the imaging right now. UTMB has some interesting constraints and she first had to find a cryostat that would cut to make the sections. So this data is actually only recorded a few months ago. So she's still in the process of cutting samples to confirm that. But yes, her goal is when she publishes to have the side-by-side comparison of the imaging and the flow.
Yeah, yes, especially also the follicular B
Yes.
cells and the germinal center that's inside.
Yep.
Okay.
So she's working on that. I just don't have the results for that yet.
Thank you very much.
You're welcome.
Well, less question, but just I work a pretty small flow core too. And so I get a lot of people saying, well, this is what I had in the box.
Yes.
So another very useful thing to ask when someone doesn't get staining is, how old is this antibody and how have you been storing it? Because I had
Yes.
someone, well, my antibody didn't stain. They had bought it in 2019 and they'd stored it at -80. So I said, well, I think I found your problem. And they ordered new antibody and wow, the staining looks so much better. So certainly with smaller labs with constraints like that is a lot of times that is part of the issue as well.
Yes. Mm-hmm.
I'll just repeat it here, although Rui's already fielding your questions online.
Oh, great.
But you can
I
comment
don't have
as
to make.
well. Is there any recommendation on calculation of dim floors using either stain index versus separation index?
Oh. Yeah, dim floors are really, really challenging. I also, you have to utilize both to kind of interpolate what you think. I also heavily use the unstained control in that case to see if the actual negative population, if the background is increasing too high, if I see that main population increasing.
Yeah, I just want to continue or comment on the same subject about, like, for some reason lots of PIs or their grad students, for some reason, they don't count the hours they used on bad research.
Yes.
Like, I don't want to buy a $200 antibody, but I'm ready to use 1 week or 1 month to do something that I can never publish. And it's sometimes frustrating.
Only slightly. Awesome, great. I'll hand the podium over. Thanks.
You introducing David? Okay. That's fine. Go ahead. You need that? Oh, well.
Sorry. All right, I'm going to introduce our next speaker, David Gravano. I'm going to go a little off script here, but he's the technical director at the Stem Cell Instrumentation Foundry at University of California, Merced. Worked on regulatory T functions at St. Jude Children's Hospital. I don't think it's, yes, you did. You earned your PhD there as well. He is an ISAC emerging leader, and that's where David and I first crossed paths because I'm very attached to the emerging leaders. I've been involved with that selection and committee for quite some time. And they were tasked, to make a very long story short, into individual groups to have assignments and do certain things. And in this case, David and Laura Ferrer, who really spearheaded this group, was to try to figure out what cores need to do to move from 1, 2, 3, 4 colors into continually higher dimensional data analysis, and what was required, what the limitations of the core were. And what he's going to talk about here is teaching a man to fish, empowering researchers in cytometry, experimental execution, and data analysis. He and Laura presented this at Cyto at 2022, and it was a very well populated workshop. Now, we're not going to run this as a workshop here, so what I asked him to do was to distill this down to a talk, and it will take into things Tuesday that we were talking about high dimensional, and also segue from yesterday's talk about cores and SRLs and how they have to keep things going. So with that, let's welcome David to the podium.
Thanks so much, David, for the introduction, and thank you for the invitation out here to speak. So what I think, we had a great introduction talk to this. From the first couple days here, you might anticipate, oh, everybody's generating beautiful OMIP quality data and running TSNE and FlowSum right now on the side while you're attending these talks and you're getting beautiful data out. And so what our group wanted to do was find out, where are we really in respect to this? Let me introduce you a little bit to my background. I've been solo staffing a university SRL at the University of California, Merced for the last 9 years now. We are a tiny core, and we serve 20 research groups, approximately 60 users. And so what does that require of me? First of all, I need to rely a lot on self-service, as I'm sure there are a lot of you in the crowd that can understand this. And what does self-service mean? It means extra training needs, because I don't know what's going on at my SRL right now while I'm here. Hoping that people are following the rules that I've taught them. So a little bit more about this working group. These are the folks that are in this SRL recognition program, emerging leader working group. David is one of our mentors in this group. And what we set out to do was really to try and assess, we're all SRL people, how can SRLs best support high dimensional data analysis? So what can we do to support people in this work? What are core facilities or SRLs? We're here to help the science, to elevate the science, right? We're here to set a good level of training. We're here to look over people's shoulders a little bit and make sure, they're not doing things like Meredith was referring to that are just nonsensical and a waste of their time and waste of everybody's time and money. But we're all also aware that, there's this paradigm shift with high dimensional data sets. And so we need to sort of find what is the SRL's role in these high dimensional data sets? How can we help people adopt these strategies? But before we can do that, we wanted to ask some questions. So some of this data is going to be from a survey that we did. And so we presented some survey results as a workshop last year at Cyto. The big questions being question number one, what is really our role as an SRL when it comes to supporting data analysis? That's not so clear cut. The second question, what are the strategies that we find are best for supporting customers in this realm? And do we have what we need to do that? Do we have the staffing? Do we have the resources? Even the expertise, right? The technical knowledge to do this. Before I go into the survey results, I want to mention there's this great program that I have to put a plug in for called the SRL Recognition Program. I don't see any Texas groups on this list of 8 labs that were recognized last year at Cyto that were able to demonstrate that they can meet ISAC's best practices that it was put out about in 2016 as a publication on best practices in regard to operations, staffing, training, a whole host of areas that SRLs operate. We're really trying to push this program. I'm one of the task force members. This was really developed by Mike Gregory and Kathy Daniels, who've been just amazing in getting this program off the ground. There'll be new SRLs recognized this year at Cyto, but we really want to encourage people that this program, we're trying to elevate everybody's science. And I think SRL is a good place to do that. Okay, so the 2 surveys we put forth last year, one went out to SRL customers, and the other went out to SRL staff members. Both went through the Purdue list, so we may be a little bit biased in who we are surveying, but we had approximately over 100 responses from each group, and a little bit about the demographics. We were mostly had people in the United States and Western Europe responding to our survey. It was very similar distribution with the customers. So customer surveys are going to be in blue. Staff of SRL are going to be in red from here on out. We got mostly academic institutions replying to our survey. We did have some commercial core facilities, but mostly academic, as you can see. And most of the people who replied were either the director or manager of that SRL. We had some faculty directors and some technicians also replying to our survey. And most of the people who took the survey as users were graduate students, researchers, but we also had some other types of scientists, postdocs, and PI and group leaders reply. So one of the questions we asked the staff members was, how many full-time staff are in your SRL? This was actually really surprising to me. 1 and 2 staffed SRL made up almost half of the population of people we surveyed. I think one of the things is when we go to meetings like this, perhaps, or Cyto, you tend to interact with the larger core groups. I think we maybe don't quite realize how many small SRLs there are in the community. But our kind of sweet spot was 3 to 5. And then the other kind of surprising thing to me was when we asked people in course, how many fluorescent channels are available on your instruments? 63% had more than 28 or more parameters on their instruments. That was, so we almost all have this technology available to us. The question is, how do we implement it well? And then another question we asked was, what do you consider high dimensional? I think the people in red, the flow core people, tend to have a higher opinion of what they think is high dimensional. Whereas our users, some of them are considering 6+ colors high dimensional panel. So let's not be too high and lofty about what we consider high dimensional. Even small panels can be considered in these ways. And so then the question became of our group, what is really our responsibility? Is this really our job, right? How much support should we be providing? And so we went to the best practices paper that ISAC put out. And, it said, SRLs have the responsibility and capacity to ensure the generation of reproducible quality data. well, is that raw data? Is that how you analyze your data? There's some interpretation there. But the document goes on a little further to mention some things that some of us were completely unaware of. The document says it should include things like, okay, general analysis theory for gating logic. We all do that. Specific software training. But then they mention data presentation, statistics, specialized modeling tools. So that these things are in that best practices paper that was published 7 years ago now. And I think a lot of us, maybe fall short in some of these respects. So we asked people, what do you guys do in your lab? What kind of services do you provide? Everybody does instrument setup, right? That's really basic. A basic course about theory of flow cytometry, panel design, data acquisition. All those things are very routine. But when we start to get down to things like at the bottom there, instrument standardization, extremely important, right? We start to get a little bit of a drop off. And then if we go on to more specialized things, and what we noticed was pretty much all the data and analysis things that we asked about were much, much lower. So I'll just point out a few examples, dimensionality reduction, clustering, normalization. We're getting down into the 30 and 40% of cores do any sort of educational outreach in those areas. And then if you get down to, computer programming tools supporting people with R and Python, almost virtually non-existent in SRLs. When we asked users, what does your SRL provide you? I won't go through this because it's really a lot of numbers, but the basic take-home message was everything was about 10 to 20% lower, especially when we got down to the data analysis tools. So maybe we think that might have been how we sampled, but I think we think we're doing a little more than maybe our users may realize we're doing, or they think we're doing. But users, the ones that responded to the survey, less than 20%, really were doing a lot of the key data analytic techniques that you've heard about throughout this conference. We asked people how you deliver these education and training modules. Most do it as part of an introductory course, or I should say half do it as part of an introductory course. Sorry, this is specific to data analysis. Most of them do it as just one-on-one or small group consultations, right? So somebody comes to you, they've got some data, okay, I can help you. I think that's how most of these things occur. But I think people are starting to work these more into their introductory courses that they provide when they teach flow cytometry. Other, avenues are in-person seminars, virtual sessions, webinars. And then we asked the same thing to the customers, and they had a similar breakdown of how they got data analysis support from their SRL. What external resources do you go to outside of your SRL? What do you recommend to people? And for the most part, people recommend, video tutorials from the software companies. That's the biggest one, about 80% there. OMIPs are a great resource. Colleagues within your institution, that's really high up on the list. But as Meredith was mentioning, your experts in the lab sometimes are a little bit dubious. And then all these other sort of sources of information are used. Where do customers feel they actually go outside of the SRL? They go to their colleagues, right? That's just the number one place they go by far. So they're, we want to make sure everybody, their colleagues, everybody is well trained in this. I think SRLs are really at a central point to do this. Do you provide any of the following resources to your users? So 82% provide some sort of a data analysis workstation. Really critical. Free access to a commercial platform, not including the instrument acquisition software. That was 64% of people. That was a big number. And the ability to have a discounted software license, 54% can do that. Then we asked people, what in the SRLs, what high dimensional algorithms do you provide assistance with? So dimensionality reduction was our highest algorithm that people provide support for. We got these categories from a review paper on flow cytometry data analysis algorithms, how we categorize these things. But dimensionality reduction was the highest. People do provide some support with clustering, but then you start to drop off, especially as you get to more niche algorithms. I think that would be as expected. 34% said they provide no assistance with any of these algorithms. What algorithms do you use to customers? 57% use none. And mind you, these are people on the Purdue list, so they're probably a little more savvy than the average SRL user, more likely. But of those that use them, dimensionality reduction and clustering are two big ones, as we've seen at this conference. Those are kind of the two big areas right now. So what areas can be improved? Where do we think we can get better? When we asked SRLs, this was pretty overwhelming. I mean, almost 70% said we need to improve our clustering support, our normalization support, our programming tool support, dimensionality reduction support. All the categories inversely related to where we already provide services. And then obviously these are all the things that we do day in and day out, like panel design and instrument setup. Nobody really feels like we need to improve much there. We asked the customers if they consult biostatistician or get any statistical support. This was generally a never. About half of them said never. The other half said sometimes, roughly. And then there were some that do consult a biostatistician, but I think this is rare, and this is kind of a pain point for all the downstream stuff, as we're all aware. We want to know if people perform power calculations, to determine sample size when they're performing these experiments. Almost universally, well, more than half say never. Some people say sometimes. And do you feel you would benefit from additional statistics training? Of the customers, 89% said they would benefit from additional statistics training. Which areas do you need more support from your SRLs? This was a little surprising that 31% said none. I was surprised by that. But then of the people who did want some more support, it was all those areas that we just talked about: normalization, clustering, all the higher sort of functions of data analysis. When we asked the SRL staff, do you have dedicated time to data analysis? People handle this in different ways. So, and people could have responded to multiple answers on this one, but a lot of people, about half the people, do it as a free service, myself included. I just want people to work with me, work with their data, and not feel like, oh, you're going to charge me $1, therefore I'm not going to ever come find you. And on a fee-for-service basis, about 40% are doing that, and as a collaborative basis as well. Is something happening? Oh, okay, thanks. Do you have links to bioinformatics support for your SRL users? No, was the most common answer. We don't have any bioinformatics support for our users. Yes, in a separate SRL was another common response to that one. And does your staff have any dedicated, do you have any full-time staff dedicated solely to data analysis? And that was an almost overwhelming no in the SRL. So we had a workshop after we did this survey, and we kind of, heard a lot of people complaining about, oh, this is too much, we got too much to do already, and we can't do this. And we tried to consolidate overall strategies into, from our workshop and our survey, things that could be helpful to people. So resources that we thought should be provided: data analysis workstations, a free access to at least one commercial data analysis platform. I think that's a big burden for some smaller labs. Site licenses if you can do it. Sample data sets and with an analysis workflow protocol. That's a big one that I'll walk you through an example in just a bit. But people really like to see relevant data to them when you're doing stuff like this. Training of users. How do we do this? Well, there should be some internal resources, such as a course within your SRL that actually teaches data analysis. But we all know that, that information doesn't last forever. People need these small group one-on-one consultations. And other people have had a lot of success with data analysis office hours. So keeping that door open, setting off a designated time where that's all you're there to do is support their data analysis. And that really reduces that sort of activation energy of them having to seek you out, make an appointment, and all the things that limit them coming to you. Some external resources people really liked were YouTube tutorials, especially from the software companies. Those have gotten a lot better over the years. The OMIPs, colleagues within the institution, current protocols. Researcher support, we really should offer this, I think, as a free service, but if you must offer it as a fee for service, that's also a common strategy that's used. How staff train each other, places like how we train staff, conferences like this, Cyto, free courses and webinars. We just need to stay motivated and stay current. And so I boil this down to what do I think my users are looking for in experimental setup and data analysis support? The first thing is simplicity. Clear step-by-step instructions and SOPs. A lot of time you'll go to a meeting, you'll see a great talk, it'll walk you through a tool or show you a tool, show you the outputs, but it doesn't show you all the steps, and they have beautiful sample data like the OMIP 69 data, right? But people really like these little step-by-step instruction vignettes. Ease of access. So this comes back to, our users a lot of the time, they might even avoid us. So imagine they're gonna completely avoid external people who are gonna, they don't want them to see their crappy data, So make your resources very accessible to them. It's already hard to get them to talk to us sometimes. So have office hours, have these workstations, tutorials on demand. I'll show you an example of kind of what I've done in that realm. Make it relevant to their application. That's the other one. Include that sample data, a full process of how they would use the tools you're supporting for them. And if you can, show them with their own data. That's really I think the ultimate goal, if you can do that. This slide was made by one of my friends, Vera, and we're all familiar with this. I've heard this multiple times already at this conference, and this is just illustrated. We want to be doing the data analysis properly so we get something interpretable in the end. The other thing I always find really, really important when I'm training people on anything and setting up experiments is order of operations. A lot of you have seen this on your Facebook feed where, most of the people, legitimately most of the people can't do an order of operations problem, right? It's the same thing with setting up experiments, doing the settings in Diva. So what I tried to do for my users, this was, solo staffed SRL, like I mentioned. I would train people. They would furiously take notes while I was doing my training, every button to click. And I said, stop what you're doing. I've made a video for you of exactly what I'm telling you. Just go back and watch it if you have any questions, if you need a refresher. So I made it for Diva, mind-numbingly simple stuff, right? Setting up a 2-color experiment, doing a 7-color compensation. But I think what resonated with people, and these videos one of them got over 100,000 views, which just blows my mind. By the way, I have a 10-year-old son who's I told him, I have a YouTube video with 100,000 views, and he was thoroughly unimpressed, right? Now, all his favorite YouTubers, a lot of whom are from Texas for some reason, and they all just have mansions and play pranks on each other all day, and they get 10 million views a day. But anyway, for flow cytometry training videos, these were a pretty good number of people actually found these valuable. But some of the comments I liked that these videos got were, I'm struggling and finding step-by-step explanation on the exact sequence of buttons to press really was valuable to people. I kind of, it also reminds me of when I look up a recipe online, I don't know if you've ever found this jump to recipe button, but it took me a long time to figure that out. And my wife eventually told me, oh, there's a button that says jump to recipe. It's great to have all the background. I want to make a lasagna, but I don't need to know the year that people started making lasagna in which region of Italy and what the ingredient, I don't need to know all that stuff. sometimes you just want to get to the recipe. And obviously we know the background of why we do these things and the theory is important, but sometimes you just need to jump to the recipe, right? So this comes back to just keep it clear, step-by-step instructions, make it easy to access. So coming back to that point, basically on all my workstations, right next to my software icon is my tutorial video icon. So literally they're almost clicking on it every time they open Diva or any other program. And then in there, keep it simple. Like I said, step-by-steps, how to refill the sheath tank, how to deal with a clog, make it as self-service oriented as possible. How to set up the fax aria. We do self-setups of our fax aria as well. And so now taking this a little bit back into the data analysis side of things. Another video, COVID started and I was at home, all my users went home. People had piles of data they had, weren't able to process. And at that time when I was looking around at tutorial videos that were out there, they were, showing you how to run FlowSum on a single sample or run TSNE on a single sample, but really integrating the whole process as the users actually use these tools was not there. Now it's starting to come out, but so my users wanted to know, how do I do this in a cross sample comparison, right? That's actually how I want to use these tools. I don't just want to look at one sample. And so another video I made during COVID was how to use TSNE and FlowSum together to really to do the process and get the data. And again, it was, people commented on the step-by-step instructions, which was really what I was going for. And then as I was mentioning now, the next level I think that core should aspire to is, can we support them with their own data directly? This is obviously a big time sink to try and handle individuals' data and work with them directly. But I had the luxury of doing it with this one group at my institution who were looking at Toxoplasma gondii infection. They were doing a B6 mouse versus a mouse that was null for a particular NF-kappa B family member, NF-kappa B ID. All they really knew about NF-kappa B ID was that they had a genetic screen that mapped to that locus. They wanted to get the knockout and they knew it lacked B2 cells, which are an innate-like B cell population, but they didn't know much more about immune responses in that mouse. And so just a little bit of the experimental background that we did. Obviously the panel design is a big part. I don't show you all the fluorochromes, but they had a really nice big B cell panel that I helped them develop. This work was done on a 5-color Bio-Rad ZE5 instrument. And they did conventional gating and analysis of the different B cell populations. And my question was, and they actually published the results. They didn't do any algorithmic analysis. And I said, can I play with your data and try and replicate and maybe find some new populations that maybe you didn't look at, right? Kind of the holy grail of this stuff. And so we restricted the analysis to just CD19 positive B cells and then went through and did TSNE and FlowSum following this very regimented pipeline that I had laid out for them. And so we generated 20 FlowSum populations. Here's our analysis pipeline, but it's similar to all the things that we've heard about in the last 2 days. And what we were able to do was first, I wanted to see, can we consistently find with FlowSum, will it cluster these B1 B cells? Did I say B2 earlier? I apologize if I said B2 earlier. B1 B cells are the innate cells that have low expression of B220, but high expression of CD19. And what was really nice was that we could get those to cluster. They form pretty much in their own independent cluster. And we could see what was known about these mice was that they lacked these B1 cells. And so very nicely, we could show that population 11 it happened to be was completely absent in these mice. Okay, so that was nice proof of what we know. And then we wanted to do the cross-sample comparison, like I was mentioning. So they had 3 mice in each group that we looked at this particular time point. They had their 3 B6 mice, their 3 NF-kappa B ID mice. And, you know, first we all like to visually just look at the pretty plot, right? And we'd like to see visually, what do we see? And we could see this, you know, this orangish population here, very apparently present in the B6 mice, not in these guys. This big yellow population here, not present in B6, but present in these guys. And some other differences if you look real close. And so I went back to their paper and I said, can our FlowSum clusters replicate your manual gating clusters? So they took B6 and NF-kappa BID mice and found that there was this defect in going from transitional B cells to follicular B cells upon chronic infection with Toxoplasma gondii. The nice thing was that we were able to get clusters that pretty nicely mimicked their transitional and follicular populations. And we could see that happened to be population 13 was the transitional, and we could generate almost the same percentages in this FlowSum cluster that they were generating with their traditional gates. A second phenotype that they identified in these mice was an issue with class switching. So they gated on the CD73 positive follicular cells and then looked at the percentage of IgM and IgD low cells. And what they found was that they had limited amounts that had class switched and downregulated IgD. And so we did something similar, and it was that orangish cluster I showed back at the beginning, but we could find that cluster happened to be number 17. Again, we were able to replicate their population data by manual gating. So that was good. You know, we were able to see what they saw using this algorithmic approach. And really my strategy for doing this was convince them that these tools are for real, right? That these things are worth your time. And then the way to do that was then say, okay, are there some populations you guys didn't look at, you know, that FlowSum pulled out that might be of interest to you? And so then we went through and just outputted a table of all those population frequencies, looked at some statistics. There were a few that were different enough to where we thought they might be worth pursuing, but one in particular, this population number 12, was pretty different in their NF-kappa BID mice. And so then we came back to the heat map and wanted to see, well, what is that population? The really defining feature of that one was it was GL7 high, but there were some other defining characteristics of that population. And so when I see these heat maps, as flow cytometrists, we don't like to average things, right? Like that's kind of against our nature is averaging because we're a single cell, you know, analysis platform. So I always take these heat maps and I need to look at all the pairwise plots of these averages in the heat maps. It's because sometimes what we're all probably aware of, or people who run these algorithms, is that you don't get beautiful clustering, right? Not everything's positive, but the heat map might tell you it's positive. You might have a good mix of positives and negatives in there. So I went back to this population and just looked at it, and uniformly, yes, it was very nicely mimicking the heat map. And it was defined as GL7 high. It looked like it had class switch, CD73 positive. So this was probably a germinal center. I'm not a B cell expert, but a germinal center B cell population. So with that, I want to talk to you a little bit about some resources that that I find are super valuable. In addition to my own stuff, I would recommend, you know, FlowJo University has been super, they've really upped their game with their, and that's the platform I've been using. So they've really upped their game with their tutorials that have come out lately. Asia Rieger, she's one of the SRL emerging leaders that is in our little data analysis cohort. She's put together some really good YouTube videos, much more theoretical, great graphical content to them. Makes me very embarrassed by the videos I made that have no edits or graphics or anything. It's just a 20-minute walkthrough. Christopher Hall's another one in our group of emerging leaders who's doing some tutorial videos on automated on algorithms and flow data. And then the MSK group here, I love your Post-its. That's something I go back to all the time to look at techniques. So with that, I just want to acknowledge my institution, but also this data analysis working group and the investigator, Kirk Jensen, and his graduate student, Scott Souza, who I worked with a little bit on their data analysis. So with that, I'll take any questions.
Thank you, David. That was
Hannah.
great. Yeah, no, I'm just curious to know if your users, they come with a specific software that they want to use or you recommend. How is this dynamic between you and the data analysis part?
That's a really tough one because I feel like I can only support certain platforms at the moment. I just don't have the ability to do all the platforms. I have what's available to me. And as of the moment, that's FlowJo. So I do have some people in FCS Express that will, bring their high dimensional data. And that's a tricky one. I feel like I can't support them as well. I can give them a little more theoretical assistance, but the the hands-on assistance is difficult.
So great talk. I'm wondering if you can speak at all to the power of observation in a solo use flow core. So I'm going to give you a little anecdote of my own. We're 2 people, so I filled out your survey. We were in one of those major percentage points. But one thing I'll do every now and again is I'll just walk around the laboratory, and the number of times I've caught people at our analysis workstation with a PDF in hand, manually entering like percentages, frequencies, MFIs into Excel or something like that. And then me going, hey, you ever heard of the table editor in FlowJo or whatever analysis platform it happens to be? And then watching their reaction as, oh my God, the days that I've wasted. Yeah, I wonder if you just comment on that.
Oh, I mean, completely. And I think COVID taught us all, right, that being present is such a huge factor, especially in how we interface with people in the SRL. You have to be you have to be just there, right, for these chance conversations to happen and to see somebody entering values and manually. I see it too. And yeah, my philosophy is just be as present as possible.
Thank you so much for those videos. I will admit I have seen those videos before. So my question is, how much is too much handholding, right? So there's a like 1, 2, 3 strike, like, hey, I have taught you this many times. Like, how do you have like a metric, or like, do you have like, say, hey, this needs to be like, where do you stop is my question.
Well, that's why I made the video, right? Because then I just say, okay, just go watch the video. If you got problems, let me know, But yeah, it is, right? It could become very time consuming.
Yeah, like to piggyback on that, like, so there's teaching and there is like, hey, I'm doing analysis for you. Like, do you talk about authorship at all with them?
Yeah, absolutely. So I've co-authored on a paper where we did algorithmic analysis. And that is kind of an upfront conversation with people. If, hey, are we going to embark on this? Okay, there needs to be co-authorship on this if I'm really going to dive deep into your data.
Okay, thank you.
All right, I have a technical question and then a comment from online. So one technical question is, when you're doing your videos, how are you recording your perspective?
With a little stand and a cell phone set on it.
Okay,
Is that what you mean?
Yes, exactly. No, perfect. There, because I know that some people were doing for remote training purposes, they were actually doing glasses that have cameras on them.
Oh, interesting.
And they're doing that for like remote training. I'm like, oh, well.
This was a few years ago. I wasn't that advanced. I've been recording like Zoom session. When I do the data analysis stuff, it's like a Zoom recording kind of a thing.
Go a little GoPro, yeah. So there was some comments that some people were showing appreciation for your videos. They've seen them.
Awesome.
As well as there was one There's one comment on noticing that 30% are doing clustering, but only 10% are doing cleaning in there. So the people are not cleaning their data before clustering. And
Well, I think we're cleaning it. People may manually be cleaning the data. Sometimes the clustering, there was a little bit of discussion about this, I think, a couple days ago. Sometimes they may be a little aggressive or, but we are doing some form of cleaning, I think, before. But yeah.
Great. And I gotta say, it's appreciated that you're putting out content like that, because some of us, and the only other question I would have is, how do you find time? Do you work Saturdays and Sundays, or?
Well,
I mean, it's.
As I showed you, we're a pretty small SRL. So honestly, I would like to be busier sometimes. And so I occupy myself with other tasks, I guess.
Yeah. Thank you. Are there any other questions for David?
So just to comment on that, I mean, this is an integral part of what we do, right, in core facilities. So actually finding time to do that is I think, an important thing. And sometimes we forget, we don't prioritize that, and then that's just left to the side. But I think having, making sure that you have time to do this, whether you're, a busy facility or not, is, I think, very important and critical. And we're a busy facility, and we do make sure that we have time to put this kind of content out there to help our users.
I mean, I think it helps me too, time-wise, right? Because when I have these a training video of my hands-on training, that, actually saves me time in the long run by having that video.
David, thank you so much for a great talk. And I think I may steal your idea and set up at least a little click of links for people to go to at the analyzers. But thank you so much. Okay, so for people in person, we have a lunch break and an ice cream social break. So we're going to start with lunch, and then we will have ice cream social. Amy's Ice Cream will be here. For those of you that are not in person, I am sorry, because the ice cream is going to be good. Hopefully you'll be able to get something yourself on the side, and we will be back in a little, well, in an hour or two.